Hello, my name is Leonor and I'm a fellow researcher of the Ultrafast Bio and Nanophotonics research group. I work for the ExtraMed project and we are developing a SyncRGB-Film Microscope, a technology that can improve imaging of biological samples as we know it. First, let's talk about fluorescence. When a molecule in a ground state S0 absorbs light of energy equal or greater than the higher energy level S1, an electron is excited to a higher energy level for a short period. The electron will undergo vibrational relaxation to the lowest vibrational level of the excited state. Then the fluorophore returns to the ground state by emitting detectable photons. Fluorophores are characterized by their absorption and emission spectra, Stokes shift, and fluorescence lifetime, the average time that a fluorophore remains in its excited state. We use Photon Absorption microscopy as a fluorescence imaging technique. The excitation is done with near-infrared photons of longer wavelength than visible light photons, and by arriving close together in time, the energy of the two photons is summed and is sufficient to excite fluorophores. For this, we use a femtosecond laser with a broad spectrum between 650 and 1050 nanometers that can excite many fluorophores simultaneously. This microscope is combined with time-correlated single-photon counting detection for film imaging. The data recording is based on detecting single photons of the fluorescence light and determining the arrival time of the photons with respect to the laser pulses, as well as the position of the laser beam in the moment of photodetection. This results in a photon arrival time histogram for each pixel in a two-dimensional scan. Therefore, during the 3D scan of a cluster of neurons of a dorsal root ganglia of red stained with API, we can generate a 3D stack of intensity imagings presenting the integral of the TC-SPC decay trace in each pixel and a 3D stack of film images presenting the fluorescence lifetime of each pixel resulted from the fitting of the photon arrival histogram. With film, we can distinguish the different fluorophores that may exist on a sample, and it's widely used to do autofluorescence imaging to monitor cells metabolism. This technology only generates fluorescence at the focal plane, avoiding out-of-focus fluorescence. This increases the imaging depth for more reliable 3D imaging. Here we have an example of 3D imaging of Uvex in matrigel stained with API and phalladine. Additionally, this characteristic decreases photobleaching and phototoxicity generated during imaging. This technology is being developed to improve the study of 3D scaffolds for tissue engineering by studying in real time the interaction of cells with materials mimicking the extracellular matrix. It could also be used to study the development of targeted drug delivery systems by investigating the interaction of healthy and unhealthy cells with the developed materials in 2D, organoids, or even in vivo models. In conclusion, SyncRGB-Film Microscope is an improved imaging technology to further advance biological research.